give you my background, talk to you about Clostridium difficile, which is the bacteria that I'm going to talk about in this case study, talk about the experimental design, the data, and then the analysis that we do. So I'm going to go over MixOmics as sort of a case study. I know Daniel talked about it, and you're going to do that later today. And then also talk about the GLASO, which is a sparsity method that I used in this project, and a mechanistic model that I developed. So just a bit about my background. So I am a mathematician through and through. My biology is a little rough. So I did my undergrad in math. I did my master's and PhD in math. I went to Virginia Tech. I did attend a Comp Bio summer school, somewhat similar to this. It was called QBio. I don't think it still exists at the time, but I actually was able to turn that into an internship at Los Alamos National Laboratory. So I definitely would encourage the interactions and the follow-ups because we have a lot of great opportunities here at PNNL as well. And to turn this sort of into that internship. So from that, I was pipelined into sort of the national lab program. I didn't even know like what they were prior to attending grad school. So I did a postdoc in a population health department at NC State. So that was my first experience to like talking to a biologist every day. Like I didn't know what lab work was. So they like let me touch a pipette. So that was cool. I had no idea about any of it. And so I was in a computational modeling group though there. I was still advised by a mathematician. We just sat on the same floor as the biologist. That was my closest that I got. So I started my career in the national labs as a computational scientist at Oak Ridge National Laboratory. I was there for 3 years. And then I moved here to Pacific Northwest National Labs. I've been here since early 2020. I am a data scientist and a team lead for One Health and biodefense. So all of my work here is in national security, doing biodefense. That's about all I'm going to say. Okay. Okay, so this work I'm going to talk to you about today, we look at shifts and the gut metabolome and Clostridium difficile transcriptome through colonization and infection in a mouse model. So this work started during my postdoc, and it is still dragging along, even though it's been a while. But so some of this is published and some of it is to be published, but so it's work I started there. These are sort of my collaborators, Josh, Casey, and Christina. They are all still at NC State. So you can definitely grab this paper if you have questions about the code. All of it is linked up there. Yeah, any questions so far? Anybody live on the East Coast or go to NC State or Virginia Tech? Yes, which ones? Okay. Nice. Oh yeah, okay, cool. Do you know these people? Oh, really? Awesome. Okay, great. Josh is coming back. He did his postdoc, he left, he's coming back. So new faculty there. Okay, cool. So I'm going to talk about that project. Okay, so C. difficile is the most common cause of healthcare acquired infection in humans. Antibiotics, they deplete your healthy gut microbiome, and then if you're exposed to C. diff, you're going to get infected. It causes all this great stuff like diarrhea and pseudomembranous colitis and death. Your colon looks nasty if you get it. Nobody wants that experience. Okay. Reoccurrence in about 30% of the cases because your gut is just fully depleted. So it's a huge ongoing issue. We're getting better about it. Some of my facts may be a little old there, but it's still an ongoing problem. So basically what happens, you like ingest a spore. If you have this depleted gut, the spores germinate, they become vegetative cells. You get outgrowth, you have more vegetative cells, and then toxin starts getting produced, and that's what's basically wrecking your gut. From that, you get more sporulation, and you just get this doom spiral, basically. So that's sort of what happens in C. diff. And so the thing that I was super interested in was toxin production, right? So toxins function by damaging the intestinal mucosa, and that's what's causing the symptoms of C. diff. That's what makes you sick, is that toxin production. The actual sporulation and outgrowth is not the issue. It appears, so the toxin appears to be triggered by nutrient depletion. However, a few studies have explored this complex environment of antibiotic depleted host gut in vivo, right? Like it's hard. I mean, the mice are going to die in this experiment, right? It's really hard to study that. And so we wanted to mathematically study what specific mechanisms are leading to toxin production. And so that was really our goal, is what is causing toxin to be produced during the C. diff infection. Okay, so the experimental setup in this, we used mice. Casey used mice in her laboratory. They gave them 5 days of water with antibiotics, then they gave them 2 days of water, and then they inoculated them with C. diff, or gave them C. diff, or made them swallow a spore. I don't know. Okay, and so then sort of in this plot at the bottom, you can see what happens. So similar to that life cycle I showed you earlier, the vegetative cells grow, then the spores are produced, and then with the sporulation is when you see the toxin and then the disease. So in this experiment, they collected metabolomics data. They had 8 time points, 8 samples per time point, and then they did the RNA-seq. We had 3 to 4 samples per time point at 12, 24, and 30. So we have time course, metabolomics, and transcriptomics data, which is a really cool experiment. We don't have a lot of samples, which is a bit problematic. And the time points are kind of wonky, like because we do 0, 12, 24, you think it would be 36. Unfortunately, the mice in this experiment did get really sick, so the experiment was ended early. Any questions about the experimental setup? Just some details about the experiment. The mice were treated with cefoperazone and then challenged with C. diff VPI, that's the strain of C. diff. And then they were euthanized at 0, 12, 24, and the 30-hour time point. So the 0-hour time point is pre-infection. Fecal contents were analyzed via untargeted metabolomics, as I said, 0, 12, 24, and 30-hour time point with an N equals 8. And then the RNA-seq of C. diff was completed at 12, 24, and 30-hour time points. With a sample size of 3 to 4, I think we lost a sample along the way. So this is a pretty costly experiment. As a mathematician, I was like, I want more data. There are not enough samples. That's like the chronic problem. I know somebody was asking, like, how much data do we need to do this? There's never enough data, right? We're always sort of pushing those statistical limits, and that's why it's super important that we keep mathematically studying how to improve our methods, right? Because we understand it is expensive to run these samples, but like it's unfortunately not a lot of times not enough samples to really do some of the more high-level mathematics, but we're getting better on both ends. Okay, so this is the summary of metabolomics. I think at this point you guys have heard about metabolomics or already knew it before you got here. But the mathematicians, when I give this talk, don't know what it is. But basically, we ran our samples with Metabolon. We got 638 unique metabolites on the 32 samples. I think that's the punchline here. Any questions about metabolomics data? You guys have heard this now for a few days. Okay. Okay, so the missing data and how we handled it. So 9.7% of our matrix entries were missing. So you don't know if it's missing because of sample preparation error, if it's below the limit of detection, or if it's truly not there. I used a k-nearest neighbor algorithm to fill in the missing data. So there's a Doe et al. Paper that talks about what to do with missing data. And so the way a nearest neighbor algorithm works is it says, okay, all of these samples are from the 0 time point, they're probably similar. And so if a data point is missing at the 0 time point, they use the other samples from that time point. And then we looked at the 12-hour and the 24-hour and the 30-hour. So we only use the samples at that neighboring time point to try to think about what the missing value might be. So then that's how we handled missing value. Yeah, and so then for the transcriptomics, we had, 3,700 genes across the 11 samples. We had that missing sample at the 12-hour time point, but that's okay. Everything was done at Michigan. Okay, so results. Any questions so far? Good online? Okay, cool. Okay, so the current data techniques with metabolomics, I think you guys have already seen some of this earlier this week. I wasn't here, I don't know. Okay, so the first thing that we did, which is a very common approach, is just looking at random forest. And we've identified the top 50 metabolites. The pictures are sort of blurry, but I'll walk you through them. We identified the top 50 metabolites that decrease. If you remove that metabolite from the analysis, the accuracy would be decreased. So that means if this, like, you need the information coming from that metabolite to make the prediction. And if we remove it, we lose accuracy in our prediction. Does that make sense? So what we see here is the very, very top one is 5-amino valerate. And so we're seeing that that one is super important. So we find that 5-amino valerate is extremely important in what's happening there. Okay. And I think the 5-amino valerate is the big kicker here for the story and for Clostridium difficile. But you can see sort of the different colors indicate the different types of metabolites. And then let's see, green and red. So the metabolite names are labeled red if their level increased throughout infection. And then what does it say? Black if they were variable, and then green if they decreased through the infection. So green if the level of metabolite decreased over time, and red if it increased. So that's a random forest. And then the next thing we also did was a heat map showing the relative abundance of the top metabolites based on the random forest analysis. So this is called an unsupervised hierarchical clustering. And we clustered the metabolites just based on similar relative abundance. Okay, so what you see here is the stuff in blue is decreasing at so this side. This blue stuff is decreasing at 0 and 12 and then increasing over time. And then the stuff on the top we see is increasing in those first time points and then decreasing at the end. So we did that clustering based on sort of, is it increasing or is it decreasing? So this combined with the other figure, we still see that 5-amino valerate, which is somewhere in the middle of cluster 2 there, is one that has the highest predictive accuracy. And then we also see that it's acting similarly to those other metabolites it's clustered with. Okay, so this is sort of the traditional metabolomics analysis approach. So then we also looked at sort of current data techniques with transcriptomics. I know I saw this yesterday when I walked in. So these volcano plots, which show you which things are acting, highlights which ones are being changed greater than 2-fold, right? So you can see between the 30 and 24-hour time point, the genes aren't really changing in that later stage of infection. But when we look at, say, the 12 and the 30-hour together, you can see several genes are being decreased and several genes are being increased over those time points. And then we sort of did a Venn diagram. So those are like the standard analysis of transcriptomics, but it's not super informative. And what I was really interested in was toxin expression and what's happening in the host gut and what's leading to that toxin expression. And none of these current analytical tools are really getting to that. So I wanted to do some multivariate based integration and see if I could understand a little bit more about colonization throughout infection. And I used MixOmics, which you guys are going to get a tutorial of later today. And so I know Daniel was just showing some of these PCA plots. And so what you have here is you have the contribution on component 1 and component 2. You've got metabolites on the left and then transcripts on the right. Okay, and so this multivariate based analysis of the gut metabolome and shows multivariate based analysis of the C. diff metabolome and transcriptome throughout colonization and infection. The top row is the first feature, the bottom row is the second feature, and then the color indicates expression levels at each time point. right? So 24 is orange, 12 is blue, and then the 30-hour time point is gray. So we see most of that first component ends up coming from the 30-hour time point. So this is like, okay, but it still doesn't really get to the question. So we use this as sort of a down select, and then we were able to look at correlations across the transcripts and the metabolites. And so this is actually a Circos plot, which is a built-in sort of feature in MixOmics. And what this does is it actually combines the metabolites and the transcripts together, and it shows you the correlation between them. Here, blue is a positive correlation and red is a negative correlation. And then the lines on the outside sort of show you if you're seeing this, if the outside, this means 30-hour. And then when this one comes out, it's a 24-hour connection. And then when the blue's on top, that means the 12-hour. Yeah, so the outer line indicates the expression levels of variable according to each class, the time point classes. So this is okay. We're starting to combine the transcripts and the metabolites together, but it's still not really answering my question of like, why is toxin being produced? Like I'm still not answering that question. And I know you guys were asking earlier, like, can you combine genes and metabolites, things like that. And so this is one way to do it. We're still not really understanding the mechanisms leading to that. And I wanted to understand the nutrient depletion. So I started talking about this more. I was like, okay, so I did sparsity sort of with the PCA, but then I wanted to look into it even more, right? So we've got this high dimensional data, we've got low sample size. We can't really analyze all of that data. And some of it, frankly, is just noise, right? Some of it is because the mouse ate its own poop that morning. It has nothing to do with the C. diff infection, right? Like some of it is noise. And so I wanted to start looking a little bit more at some of these graphical models and some better interpretation. And so I looked into what's called a GLASO, which is a penalized method to make sparse networks with fewer entries. That only includes the most important associations. So what happens with a GLASO? Basically, it's a graphical model. You see these lines are correlations between metabolites or transcripts. Positive is usually green and red is negative, right? So they're both being upregulated together, or they're one's up and one's down, some sort of correlation there. And what a penalized method does is it takes out those weak correlations so you can ignore them, right? So in this little graphic here, you see basically the thinnest lines get removed because they're weaker. And it's like, okay, well, let's just look at the stuff that looks really strongly correlated because we have to pick something, right? So this is sort of the idea behind the GLASO. It's just a penalized function, and it just removes the weakest correlations. It is a partial correlation matrix, so it removes correlations of confounding variables as well, versus just a normal correlation matrix. But the GLASO is cool. People use it. I don't think we're going to actually do a demo of it, but it is something that people are using with omics data. Okay, so sorry, this is supposed to be a lambda equals 0.08 up there. It didn't quite come through, I guess. But so this is the same network of metabolites. Now, everything we were just talking about with the correlations in between, Lisa calls this a hairball network because it's still not really useful, right? Like there are all of these methods that aren't really getting at interactions, right? And so you can sort of see the one thing that's interesting here is as you increase the penalty, like the number of nodes will go down. So this is just one idea of lambda equals 0.08. As you increase that lambda, the number of connections and nodes you're going to see are going to reduce. And so this is like, okay, but it's still a lot. But I was still really interested in like the whole time. I'm like, okay, what is driving toxin? Like what nutrients are connected to toxin? And so I just went ahead and like through my transcripts. The text thing is a rendering error. Sorry about that. I went ahead and just threw the transcripts in with the metabolites. I looked then specifically at what is correlated with TCAD and TCADA and TCDB. Okay, I think I added a letter in there, but Okay, and I wanted to see what is correlated with those toxin expression genes. And what I found was the strongest correlation was coming from 5-amino valerate, which is actually what we saw from that random forest plot like 15 minutes ago. So I was like, okay, 5-amino valerate seems like important, right? And then I also found out proline was there as well. And so I was like, I'm a mathematician, and I'm like, I don't know what proline and 5-amino valerate are. Do you guys know what those are? Have you heard these words? Do you know anything that they do? So I started just like Googling things. I'm like, what's proline? What's 5-amino valerate? Okay, well, turns out that when like an electron donor, which in C. diff is oftentimes leucine, interacts with a protein, it actually then breaks down, like it breaks apart, and it makes 5-amino valerate, which is what C. diff eats for energy, right? And so that's actually where that toxin production is coming from, is actually from this breakdown of proline into 5-amino valerate, which is known as the Stickland reaction. Have you guys heard of this Stickland reaction? Okay, not C. diff people, but if you are, everyone's really excited about it in like the last 3 to 4 years. Everyone's like, oh my God, the Stickland reaction. So it was really cool that I like found something that was like actually a mechanism that was like a thing, right? It wasn't just like a random correlation or connection. I was like, okay, this is a thing. So I was like, okay, so what do we know about the Stickland reaction, and like what sort of actual like increasing and decreasing interactions can we figure out? So I took this and I actually developed what we call like a mechanistic model of the Stickland reaction. So this is a system of ordinary differential equations. They somewhat became popular with like the SIR model when everyone started talking about COVID, but like they've been around for a really long time. And so I actually made a mechanistic model of the Stickland reaction, okay? And so this is basically just saying you need the electron donors to interact with the proline to make the 5-amino valerate, and all of that is going to increase toxin, right? And so through, backing up through the glasso and inducing sparsity, I was able to actually identify this process and mechanism leading to toxin production. And then I, made a math equation of it because that's who I am. I'm a mathematician. Okay, so I took this mechanistic model, and then we're kind of just running out of time, and this figure is super blurry. But I used it and I fit the parameters using this thing called an MCMC, which is Markov chain Monte Carlo simulation. But the kicker here is I actually fit the parameters and I fit the metabolites to a mechanistic model. And so then I was actually able to sort of look over time. So these are actual data points of metabolites of the electron donors, the proline, and the Stickland, or the 5-amino valerate, and then fit that to sort of understand the toxin. So the data is super noisy, but the main things that you see here is as proline decreases, 5-amino valerate is increasing, and that's also leading to increase in toxin. So this is kind of cool. And then you can actually start looking at how these different reaction rates in your mechanistic model affect toxin production. And so like one of the things that we found was like if you were actually able to remove that electron source, you're not going to be able to fuel the Stickland reaction, and so you're not going to be able to produce toxin, right? And so that's sort of coming from this. So the top one here is the electron source graph. And so, wait, did I say, wait, okay. So if you add some electrons, you fuel it, and then you can like over electron it, and your toxin is going to decrease. Same thing with the proline source. Some proline is going to fuel it, and then you can basically just wash it out. And then if you decrease it to the left, you're going to see also less toxin as you decrease it. So you can sort of look at the reactions there between the mechanisms in your mechanistic model and see how that's going to actually affect that proline production and the 5-amino valerate, and then reduce the toxin in the long term. Okay, So conclusions here, sparse graphical models are suitable to develop a mechanistic model-based approach of omics data. And the Sticklin reaction may be influential mechanism in driving toxin production. And then our model would suggest that adjusting the inflow of metabolites used in the Sticklin reaction could be used as a primary intervention strategy.